All right, this is the final section of Chapter 4, where I wanted to talk about the last two Baltimore classes of viruses. And these last two are arguably the most complicated in terms of their replication. Um, so these are the RNA viruses that reverse transcribe in Class 6 and the DNA viruses that reverse transcribe in Class 7. And I'm going to focus primarily on Class 6 here. I will give you a little bit more info on Class 7. Um, but the RNA viruses that reverse transcribe are also called retroviruses. This includes HIV in the family Retroviridae. And so these retroviruses do have positive sense RNA genomes. Interestingly, the virion actually comes packaged with two copies, even though only one gets used. But despite the fact that they have that positive sense RNA as their genetic material, uh, the genetic material itself is not infectious. And that's because it does need some of the other components of the capsid in order to be able to complete the replication cycle, primarily the reverse transcriptase and an enzyme called integrase. So reverse transcription is carried out by this enzyme called reverse transcriptase, and as that name implies, like transcription would be going from DNA to RNA, so reverse transcriptase will reverse that process and make a DNA copy from RNA. And so what we see in the retroviruses is once the RNA genome is inside the cell, reverse transcriptase makes a DNA copy, and then that DNA copy integrates with the help of an enzyme called integrase into the host chromosome. And that reverse transcriptase is a heterodimer. It's made out of two different separate polypeptide chains called P51 and P66. You can see those illustrated here, where P66 is the green, blue, gray, basically the top half, and P51 is in orange on the bottom. And reverse transcriptase really has three main functions. So it can act as an RNA-dependent RNA polymerase, or sorry, RNA-dependent DNA polymerase. So basically producing DNA from an RNA template. Can also act as a DNA-dependent DNA polymerase, so making DNA from a DNA template. And it has RNase activity, meaning it's capable of degrading the RNA portion of a RNA-DNA duplex. It does not proofread, even though it has this DNA polymerase activity, and it's much slower than normal cellular DNA polymerase. So I'm just going to go back here just for a second, just to the flow chart. So again, this positive sense RNA genome gets inside the cell, reverse transcriptase makes a DNA copy that we're going to call the complementary DNA or the cDNA copy. That then gets integrated, and I'll show you how this reverse transcription takes place here in just a second. Once it's integrated, that's called proviral DNA, and the virus may stay in that proviral state for a long time. So with HIV, at that point, you would be considered HIV positive, right? The HIV genome is part of your genome, but you may not get sick right away because there may not be much active transcription and translation of viral proteins. But at some point, that will typically ramp up. So you start producing viral proteins and enzymes. We'll start actively producing positive sense single-stranded RNA copies of the genome and producing new infectious virions that are then going to spread. And in the case of HIV, of course, the virus is preferentially infecting white blood cells, so it ends up decimating your immune system. So I wanted to show you what the genome of a typical retrovirus looks like, and this is going to tie into the process of reverse transcription. So at either end of the genome, you have these redundant sequences labeled with R, and then you have sequences that are unique to either the 5' or the 3' end, labeled U5 and U3 respectively. And then you have a primer binding site that will be bound by a tRNA, so a cellular tRNA that's going to act as a primer. And here's what reverse transcription looks like. So there's a lot going on here, but it's really quite elegant. And so I wanted to just kind of briefly walk you through this just so you have some idea of what's going on here. So keep in mind this is reverse transcription, right? Making DNA copy of, double-stranded DNA copy of that single-stranded RNA genome, starting with the primer. So basically everything in light green here is going to be DNA that's being synthesized. So we start with that tRNA primer binding to the primer binding site. Reverse transcriptase can elongate from the 3' end of that primer, so basically making a DNA copy of the redundant and the unique 5' sequences there. And then it's got that RNase activity, so it degrades the part of the RNA that corresponded to R and U5. And then we get this phenomenon called primer jumping, where now, because we have this rDNA sequence that we just produced that will be complementary to the rRNA sequence over here at the 3' end, this whole elongated primer now can jump to the other end of the RNA and be held in place by a complementary base pairing in that R sequence. So now our 3' end is available at the opposite end for elongation, the opposite end of the RNA. So again, reverse transcriptase can build off of that 3' end and basically make a DNA copy of the rest of the RNA. And then again, RNase activity will kick in and it will chew up all but this small portion of the original RNA, which you can see here, basically right in the middle. The 3' end of that RNA will now serve as a primer, so we can start building the second complementary strand of DNA. Can extend off that 3' end. Once that's been extended, we don't need the primer anymore, so it'll be degraded. And then again, we have a similar jumping as we were talking about with the primer jumping. So this primer binding sequence can now jump to the opposite end of the DNA. So remember we had our original primer binding sequence here. Jump to the other end and provide that 3' end for elongation yet again. And we will end up with a 3' end here that can also be elongated. So eventually we end up with this full double-stranded DNA copy of our original RNA. And as we discussed, that is then going to integrate into the host cell chromosome with the help of the integrase enzyme, which as far as researchers can tell, basically picks a random spot in the cellular DNA in the genome and facilitates this cut and splice essentially that is going to result in now the HIV DNA being integrated into the cellular chromosome where it will now be considered proviral DNA. And at that point, it's just like any other cellular gene in the genome, right? And as we discussed, some point down the road will be transcribed and translated at a very high level to produce new viral proteins. All right, so the last class I mentioned was class 7, which is the DNA viruses that reverse transcribe. And that might seem confusing, but I'll try to explain here in a second. Why would a DNA virus need to reverse transcribe? But there's really two families of DNA viruses that carry out reverse transcription. Calypnoviridae, which is a plant family, and Hepadnaviridae, which includes hepatitis B. Of course, hepatitis is an inflammation of the liver, which can be a serious disease in its own right, but can also lead to liver cancer. We'll talk about that in later chapters. So pretty serious virus. And we just talked about retroviruses and how they're using reverse transcriptase. So what are these DNA viruses doing with it? Well, I'll show you a little bit more information on what their replication cycle looks like. But in a nutshell, they begin with this partially single-stranded, partially double-stranded DNA genome. It's pretty unique in that regard. And that will get converted into a fully double-stranded, what's called the covalently closed circular DNA. So basically fully double-stranded. Uh, and that can then be used for transcription translation of viral proteins. But this will also be used as the template to form what's called the pre-genomic RNA through essentially transcription process or transcription-like process. And this is where the reverse transcriptase comes in now, is using that pre-genomic RNA as a template to basically regenerate our partially single, partially double-stranded DNA genome. So we're going from RNA back to DNA. So it's really for that that nucleic acid replication step essentially is taking advantage of that reverse transcriptase. And so I mentioned partially single-stranded, partially double-stranded genome. What does that even mean? Well, it looks something like this. So you can see, there's portions here, like on the right side, where this is completely single-stranded DNA. And then we've got portions like this on the left side that are completely double-stranded. So very unusual. This is technically termed the relaxed circular DNA, or rcDNA. And as I mentioned, one of the first things that will happen is that will get converted into fully double-stranded form called the covalently closed circular DNA. And that's not going to integrate into the host chromosome. It's maintained as what we call an episome, so separate from the host chromosomes. And then the cellular RNA polymerase can transcribe viral messenger RNAs. And so I don't want to go into all the details here because I already probably overloaded your brain going through all the reverse transcriptase steps in the previous class. But let's just suffice it to say that the replication steps in this class are also quite complex and also quite elegant. But I'm not going to hold you responsible for these. I'd rather you focus on the retroviruses for now. But if you're interested in digging in, you do have some more information here and in the book. All right, thanks for hanging with me. That is the end.